Good morning, good afternoon, good evening, everybody. I'm Carla Roslin. I'm a co-founder of the Global IMMUNOTALKs, and it's an absolute delight today to introduce our global immuno speaker, Dr. Irina Udalova. Irina is the head of the Genomics of Inflammation Laboratory and the Associate Director in Data Science at the Kennedy Institute of Rheumatology at the University of Oxford. She's a molecular immunologist with a multidisciplinary research background. She did her postdoctoral work at the Wellcome Trust Center for Human Genetics at the University of Oxford, and then first established her own group at Imperial College London. During this time, she focused on transcriptional regulation of the inflammatory response, and her own laboratory led groundbreaking discoveries of the transcription factor IRF5 as a master regulator of pro-inflammatory macrophages. I very much remember vividly, Irina, when you were making these really wonderful discoveries, which told us a lot about macrophage polarization, plasticity, and how you could start targeting IRF5 dependent mechanisms to impact the inflammatory response. After her initial time as an independent scientist in London, Irina returned to Oxford, where she expanded her own program to focus not only on macrophages, which I think we're going to learn more about today, but also on neutrophils, and particularly the transcriptional heterogeneity and the multitude of functions that now we know neutrophils have, in large part thanks to Irina's work. On a more personal note, I met Irina a while ago when we were younger, probably about 15 years ago, when we were both assistant professors. We were very fortunate to be funded at the time by the American Asthma Foundation, who brought together many immunologists across the world from different areas of expertise. And as somebody that is from the same generation as Irina, it has been my privilege and my pleasure to see her advance so nicely in her career and to contribute so importantly to our current understanding on the biology of myeloid cells. I think we're in for a treat. I very much look forward to Irina's presentation of her global IMMUNOTALK entitled Biochemical and Biophysical Regulation of Macrophage Development and Function. Irina, thanks so much for accepting our invitation. It's a pleasure to have you today.
Good morning, Carla. Thank you for having me, and it's such a pleasure to see you again online. We just had a conference together last week.
Yes.
At the Gordon conferences in phagocytosis in New Hampshire. So it's really nice to catch up again. And yes, I'm really glad to be here.
It's always nice to see you. We're delighted to have you. As you know, we love to hear and learn more from our global immuno speakers. So we have a question for you today. I know you and I were talking, we still feel young, but what would you say to your younger self if you were to give some mentorship advice?
Yeah, it's a good question. I think, I would say to myself as well, as much as I say to my current, early postdocs and students, is try to go for your own project and your own fellowships as early as possible in your life career. Not only is going to help you with your building of your CV and your presentation and visibility around, but also it trains you in terms of how you handle research questions, how you handle the budget that has been dedicated to answering those questions, how you organize, how you make the logistics. I think it's an invaluable experience. So the earlier you start, the more chances it will be that you will be successful later on. Excellent. That is my big kind of advice. And there is a small advice, it just reminds me, because we came back from the conference that was exceptionally well attended, but also very active. There was lots of time for discussion, and lots of PhD students and postdocs were quite vocal, and asking questions, not feeling shy and so on. So the little advice that someone told me, and then I'm always passing to my people, when you come to a conference, try to ask the question, to the first or second talk of the conference. As soon as you do once, you're going to be unstoppable. Because often we feel a bit shy, a bit timid, you know, should I ask, should I not ask? Is it a clever question? Is it not? I think it's not the point. I think you need to be confident and you need to be able to ask. So try to ask early on at the conference and then you will be fine and everyone will notice you.
That's wonderful. Thank you so much for that advice. So important to get an early start and try to, you know, if we apply, then you have a chance, right, to get your grants. If you don't, you obviously won't get them. So thank you so much, Irina, and we very much look forward to your Global IMMUNOTALK. Let's see if you can share your slides.
Right.
That's perfect.
You see it, yeah?
Okay, we're all ears.
Okay, thank you. So today I will be talking about one of my favorite cells, which is macrophages. Another one is, as Carla said, are neutrophils. So, and every time we do something, you know, when we study the cells, we find something new about them. And this time around, we kind of ventured into the field of biophysical regulation of macrophage development. And that is something that we are very much novices in and very much like to develop further, but I'm very excited about it. So it all starts with, let's see if I can blink. It all starts with our interest in synovium or the joint. This is like comes from the fact that I am actually in the Kennedy Institute of Rheumatology, where studying the joint inflammation or joint destruction is one of the main topics of the whole institute. In terms of the macrophages in the joint, there's probably less studied cells compared to some other organs. And what we know from the very early on, from the old seminal papers on the structure of the synovium, is that there are some macrophages that integrated in the synovial lining, and there are some macrophages in the sublining. What they are, what they do, it has been a quest for a few labs in the field, not that many, but around 2020, there were 2 publications that started looking with all the new technologies, such as single-cell RNA-seq analysis, at the heterogeneity of macrophages in the joint, just collecting everything that is there and see what sort of phenotypes they present. And in the mouse, the 4 major populations have been described, highlighted here. And so in the humans, there are about 4 major populations that have been described. The question is like, there are some markers that those populations are expressing, but what they are and what they're doing has actually caught our attention around the time of, not sure why I'm, okay, not changing slides well.
Sometimes in the bottom left.
Okay, yeah, maybe I'll
there. That's perfect.
It's changing. It's probably because of that. Sorry. Oh, I disconnected. Okay, I'm sorry about that. If I'm still, Carol, can you hear me?
Yes,
still online?
yes,
Okay.
You're still online.
I'm just
a
trying
beautiful
to find
image.
a way of clicking the slides. Yeah. So the beautiful image here indicates the joint structure with the femur and the tibia around it, and the kind of the synovial lining here with the sublining and fat part there. And if we label those 4 macrophage populations in different colors, identified by the markers that have been there, you can see that they're spreading around. And let's just focus on the more specific areas where they are. So you can see that there are 4 different areas here, and they have like different patterns of presence. Some of them are also detected in the bone marrow that we can conveniently see here, but some of them are only synovial related, like here, which is the lining macrophages. And so there has been some interest in all those populations, and we now know a little bit more about each of them. So let's start with the first one, which is one labeled by VSIC4 CD68 macrophages, which are the synovial lining macrophages. So the work from Christina Zek from my group showed that these macrophages are the ones that are recognizing the immune complexes in the models that we are running when we induce the inflammation. For example, here you can see do you see my pointer? Let's see here. Okay. You can see that they're located in the lining layer. They're the ones that are filled up with the methylated BSA and immune complex with it, which is a model of inflammation we are running, is induction by that immune complex. And also, these macrophages are the one expressing the first signal for recruitment of neutrophils that seem to be coming specifically into that area of the synovium in the lining at the early stages of inflammation. And later on, they just spread around the whole synovial sublining and part. Some other people also indicated, in addition to being able to recognize initial onset of inflammation and the challenge, the same macrophages are quite important later on when after the inflammation is resolved for rebuilding the lining. So they have two different roles. First, sampling around and seeing what's going on and what's happening and calling the neutrophils in if something needs to be cleared up, but also being immunoprotective at the later stage, rebuilding the lining and closing that insult point. Another population of macrophages, realm alpha CD68 here, localized in a different location. They are in the sublining area. And what Barbara Schoenfelder from my group, a PhD student, found that they actually position around next to the vessels in the sublining, and they are the one expressing high level of CCL2 here. So the next to the vessels expressing CCL2. And in the early stages of inflammation, again, in the same models that I described previously, they are the ones which seem to be important for recruiting monocytes to the lining. So we have like this interesting duality. Neutrophils go in one place, monocytes go in another place at the very early stages, and later on everything mixes up and it becomes like filled up both lining and sublining. So the functional consequences of it, it's not very clear yet why they need to be two different areas for recruitment of different cells, but that is an observation that we're still kind of trying to dissect further. Another beautiful study comes from Mena Klatforce about the third population of macrophages in the joint, labeled by MSC class 2, which are found predominantly in the interface between the lining and the sublining layer, and they form complexes with no receptor neurons, like she called them the sentinel unit, around fenestrated capillaries to block, to defend the synovium from circulating immune challenge, so to stop more immune complexes coming into the lining. So this is, you can see how they kind of make the plug on the fenestrated vessels. That leaves us with the last population labeled by aquaporin 1, and this one have not been previously looked at. So this is the main topic of my presentation today, and our current interest in all the biophysical and immunomechanosensitive sort of properties of the joint. So how we came about it, where they are. They're very similar to where the lining macrophages are, but slightly different. So they're a little bit closer to the meniscus and they occupy the specific area there. You can see on two different presentations. And if you start looking at the 3D kind of location of lining macrophages VSIC4 in the cyan and aquaporin positive macrophages in the magenta here, you can see that they kind of come close to each other, but you can distinctly see them too. And in fact, if you now project on 2D, our 3D rotation of the knee, you can see that those macrophages are largely located in mechanically stained areas of the knee, close to the patella tendon, close to the menisci, and close to the cruciate ligaments. Those areas are normally mechanosensitive. And how do we know that they are mechanosensitive? Is there is this molecule called YAP1 that normally labels the mechanically active areas in the tissue. And you can see here, it's in a cyan, that it kind of close to the aquaporin positive macrophages in similar location. And if you do the intersection of a triple labeled cells like aquaporin 1 positive, CD68 positive, and YAP1 positive in green, this is basically where they can be. So it's likely that these macrophages are located in the mechanosensitive areas. So what is it about this mechanosensing? In fact, the joint is a very mechanosensitive organ. You know, it's both macromechanosensitive because it moves, and also micromechanosensitive as other tissues, for example, in response to inflammation or some kind of a stress when the tissue changes its parameter. So mechanical activity is important for maintaining the function of the joint. This is like why the joint is so flexible. There are some indications that patients with immobility, for example, after spinal cord injury, display cartilage synovial. So if you don't move, you kind of stop being so functional. But the effect of mechanical activity on the function of synovial macrophages has not been studied. And so Barbara Schoenfelder, like brilliant PhD student in my group that already looked at the previous, another population of macrophages, I mentioned her, actually took this colossal project in her stride and developed our kind of understanding of what might be happening in this system and what these macrophages might be doing on a functional level. So the first thing we asked, or the first thing she asked was, is movement is really important for their health and, you know, maintenance of these macrophages. And in fact, if you stop the movement of the joint by doing the sciatic neurotomy, which restricts the movement, you can start seeing that the number of those macrophages in this mice subjected to this operation, surgery is reduced. You can do the opposite. I'm sorry, it's the other way, which is putting the mouse on the wheel, so enhancing its lifestyle and making them to move a little bit more. And in that case, you can see that there is an increase in the number of those macrophages present in the joint. So do they come to the joint early? Do they come out of being like monocyte differentiating into macrophages in the tissue? And so to answer that question, we teamed up with Dr. Rebecca Jantic, who is in Edinburgh, and looked at the aquaporin positive macrophages in the knee joints of newborn mice, and we could detect them, which is an opposite to the lion macrophages that actually is not really present until mice are born and start moving. And Rebecca is developed a very beautiful story about at what point we start seeing different macrophage populations in the joint. We can also look, so they are embryonically seated, they are present in the embryos. But we can also look at a different way, whether they are, whether the main source of them is monocyte derived versus, you know, this tissue resident macrophages. And for that, we use this quite popular report. The strain is MS4A3 CRE-DD tomato that labels the, monocytes and macrophages derived from monocytes. And in fact, you can see that there is quite little of the red color in the lining area. And if you start counting the cells that are lining aquaporin positive and red, you can see that there are not many of those. About 15% of the lining macrophages are monocyte derived, which is consistent with other work identify those macrophages in their origin by other means, but very little of aquaporin, even less, about less than 10% are monocyte derived. So we think that in the steady state, at least, the major contribution is from them being embryonically seeded and surviving and doing something in the tissue, possibly proliferating and renewing themselves, which is in fact the case. We asked whether they can proliferate, and we used that EDU labeling of the cells, and we can see that some of them are in fact proliferating, having positive EDU signal, and lots of EDU signal, obviously, in the bone marrow here that you can see as a kind of part of this image of the joint. In addition, we could now look at those cells in vitro setting, but they can take them out, or we can use the model system of Hoxb8 cells, which are the immortalized progenitor cells that you can differentiate in vitro into macrophages. or neutrophils. There are different clones. In this case, it's the macrophages with GM-CSF or MCSF exposure over 5 to 7 days. And so in these cells, you can also generate various genetic modifications. So we produced wild type and aquaporin knockouts in these cells. And we could see that indeed the wild type cells can proliferate at the day 2 progenitor level quite well, and also quite well at the day 5. And the aquaporin deficient cells in this case are less capable of proliferation. So what is that population of macrophages for? So we know now it's responsive to the movement of the joint, is located in the mechanically strained areas, but what are they for? Why are they there and what they are doing? So based on the profiling of single cell macrophage populations, like using the dimensional reduction plot, trying to do the trajectory of their development, they kind of located in the middle, in possibly before becoming the proper linear macrophages marked by VSIG4. So similarly, if you start looking closely at the joint sections, you can see that there is kind of a progression in the lining area between the aquaporin positive macrophages in the meniscus through the double positive aquaporin and VSIG4 cells to single positive VSIG4 cells. So we kind of hypothesize that maybe the function is really being a precursor reservoir of macrophages to form the lining when it's needed, to substitute the cell there. So we started playing with that idea, looking at various possible ways how we can do it. And one of them was using the confetti mouse, where you randomly label cells with different colors, and then you can see whether the cells of the same color are located next to each other. So that would indicate that the cell that was, say, proliferating and differentiating at the same time from the same kind of clone of cells next to each other is more likely to be one precursor of the other. In fact, here there is an example where the two main colors we see, it's yellow and green here for the confetti mouse, and then imposed on the magenta color and cyan color of VSIG4, you can start seeing that some of the cells are labeled by both the yellow and then magenta and then the VSIG4, and there are some other examples of those. With quantification of the adjacent cells being of the same color, both aquaporin and the VSIG4, of different color, aquaporin and VSIG4, you can see that the more likely to be together indicates that there is a possibility that one becomes another in the right conditions and the right sort of niche signals of the lining. Another way of kind of looking at it in a more sort of in vitro setting is isolating the aquaporin positive cells, and there are only a few of them, it's like 100, 200 cells in the joint. Therefore, it's quite a tedious, like difficult experiment to do, but it's possible to culture them and to support the culture by hyaluronic acid-rich hydrogel for sort of more physiological conditions representing what's going on in the synovium. And if those cells are kept in culture and subjected to the supernate and also extracted from the culture of the total synovial cells, after a certain time, they can become VSIG4 positive expressing macrophages here. So meaning that even in a vitro setting, when we isolate them, there is a chance, but if you put them in a special condition, and I will refer to it later why it's important, they can start expressing VSIG4. So what is it all about? What is this aquaporin? So one thing to mention is that those cells are also expressing CD44, which is the receptor for hyaluronic acid. And in fact, the area is quite rich in hyaluronic acid. That's why we use it also for the hydrogel system. But also this is something that is important for structuring the water molecules in a certain way, so it would be easier for the aquaporin, which is a water channel, to recognize and transmit them. So in addition to this passive diffusion of water in the cells, about 10-15 years ago, there was a big discovery, and a Nobel Prize for it, of aquaporin channels, like active channels of transmitting water in and out of the cells. And this is one of them, of the family of, I think it's up to 15 or something like that, aquaporins. The function of which and what they do and what it means for the immune cells, it's still very much under investigations. And aquaporin 1 has not previously been actively associated with macrophage biology. So we started looking into it not only as the marker of these mechanosensitive cells, but actually what it might be doing there. And is it important really to have this channel highly expressed on the population? So the first thing that we do is more like immunologically relevant rather than biophysically and biochemically relevant, is seeing what happens to the lining layer in the mice that lack aquaporin in macrophages. So we generated a cross of lyse M aquaporin flocks mice and deleted aquaporin specifically in macrophages in the joint. And we could see that in that case, instead of having these aquaporin positive macrophages uniquely, not that much of the VSIC4 in the meniscus area, now this area is populated by other types of macrophages. So there is something going on in the shifting of macrophages around. So they start adopting a different space to what they need to be in a norm situation. So they move into this area. So it's a lot of localization of lining macrophages. And if you start looking at the more sort of zoom out way at the whole lining structure, you can see how well it's presented here. This is like the control mice, and you have a quite a beautiful lining of VSIC4 positive macrophages. And that lining is disrupted if you don't have the aquaporin in this small population around the meniscus, which is quite a profound effect, considering that it's not that many macrophages and it's just one specific molecule water channel there. Now, does it have anything to do, or does it play any role in the inflammatory situation when we have inflammation such as rheumatoid arthritis, say? So in the mouse, we have different models of inflammatory arthritis or articular inflammation. And one of them is a very simple one that we're using is when we immunize the cells with a methylated BSA in the presence of complete CFA. And 7 days after, we challenge the knees. One of them is challenged with PBS as a control, and another one is challenged with the same antigen, methylated BSA. And then we start looking through the progression of inflammation, which is the peak of it at day 2 post-injection of methylated BSA, and the resolution occurs between day 7 and day 14. And so we can follow the mice there, also looking at the swelling without actually touching the mouse, and just with a caliper, you can look at the size of their knees. And you can see that the absence of aquaporin, which is here in the magenta here, increases the swelling. So the inflammation is enhanced. The area on the curve also shows quite a strong induction. If you start looking at how the mice step on a platform, and they can lean on one or another leg, and if the one leg is inflamed, they don't lean on it so well. You can see that the leg that has been challenged with methylated BSA in the aquaporin deficient mice they don't like to step on it that well. And also, from the immunological parameters, you can see like the measure of inflammation would be infiltration of neutrophils, say, into that joint. You can see there is more neutrophils infiltrated in the joints of the lyse M aquaporin knockout mice, which means that altogether with the disruption of the lining that I was showing you, and we're also seeing it here, it's probably likely that it's kind of the resolution of inflammation is not occurring as effectively, and the lack of the sealing, the lack of the lining rebuild is causing this progression and maintenance of neutrophil recruitment and swelling and what's not there. So again, going back to the in vitro model of Hox B8 system, and trying to see whether, in addition to just not closing the lining, the type of macrophages might be different as well. You can look at the differentiation of those macrophages from the point of view of the expression of surface marker. And you can see that the lack of this water channel changes the buildup of macrophage itself. So it goes more inflammatory here, comparing to the more sort of restful macrophage in a normal stage, which can also contribute to how inflammation is progressing and how early or late the resolution can come about. Another interesting part here is when we look at this system, and you can see it's just in the dish without any further support that I previously introduced with hydrogel matrix. In that particular system, we never get the expression of VSIC4 on these HOXB8 macrophages, despite of them presenting quite a good range of markers. But they can never get the VSIC4. So we've been wondering what was missing in this system, why they never get the VSIC4 expression. And then we came about with this idea, like going back to the original, like why, the cells are in these mechanostrained areas, what it means, do they need a further impetus, do they need a further stress, do they need some mechanical pressure to actually become the type of macrophages they need to be. So yes, what is missing, we thought was maybe mechanical stimulation. And then we started looking at the cells and how they are in situ, and when we just extract them out of the joint and measure the size of their nucleus, and we can see they're quite compressed. So the nucleus is about 4 micrometer in size. At this point, we got together with Matthew Peel and Anna Maria Lennon-Duminil, who been working on the production and use of various chambers, in vitro chambers, where you can start pressing the cells and you can start measuring whether that pressure on the cell is actually constraint of the cell is actually making them to behave differently, immunologically and physiologically. So we argued that the normal size of macrophages, or HOXB8 we have for the nucleus and soil, is about 9 micrometer. We don't press them with that, but when we have this 4 micrometer size, it's when they need to be really compressed. And so Matthew Peel and Anna Maria has given us those chambers in which we can generate the conditions for either keeping the macrophages largely intact or quite strained, constrained in that. So we started looking whether that would change their biology. And in fact, it was quite spectacular, because specifically for some other markers as well, but specifically for this VSIC4 expression, we can only gain its expression after we press the cells to 4 micrometer size in their nucleus diameter. And in fact, we couldn't get that working for the aquaporin deleted HOXB8 macrophages. Not all the markers responded to the stress, like some classical macrophage markers, CD206, were completely oblivious to this strain. But some markers, very specific for the joint, were really responsive, not only to cells having a build-up, of proper build-up, but also having the biophysical parameters needed for them to become a certain type of macrophages. So the first conclusion that I want to make, this part of the story based largely on the in vivo and in vitro data, is aquaporin positive macrophages localized at the mechanically strained areas of the joint. Their maintenance depends on healthy mechanical loading of the joint and is impacted by mechano-inflammatory arthritic pathologies. They are embryonically derived, and we think they are precursors of the lining macrophages. They adopt lining macrophage phenotype in the lining niche, or when we press them, when they're outside of the niche, and they require aquaporin and compression. We don't know yet for sure what transcription factor controls this differentiation, but we have some candidates that may include some IRF molecules from the families that I've been working on previously, or quite a lot on. And the lack of aquaporin expression on these macrophages leads to the exacerbated inflammation. So this is a kind of the schematics that we are going on. So they are here, they're proliferating, they may be overcrowded and going out of that niche, becoming the different types of macrophages, and the compression is needed here. So how do they get out, and why do they get out, and what regulate that getting out situation? So this is again the chamber experiment, and this time we're looking at the live cells and their motion and migration. So you can see here that not much is happening without any compression, and after being compressed, after being pressured, the yellow cells became much more mobile. They want to get out as aquaporin knockout cells, which are red here. They are still not that mobile. So the compression is needed for the cells to get out, to move on, and the aquaporin is needed for that. There is one classical pathways that people were looking at, which can kind of work as a sensor of that critical stretch on the nucleus achieved by either confinement or swelling. And it's quite important that it can also be the swelling, because obviously the aquaporin 1 being a water channel is very intimately linked to the cell being able to become bigger and smaller depending how much water is in it. But both pathways kind of come together on the activation of one specific enzyme, which is located as activated form in the nuclear membrane, and it's called phospholipase A2, which is one of the important enzymes for producing bioactive lipids such as eicosanoids from the arachidonic acid. And those eicosanoids like prostaglandins, like lichens and so on, they're important for governing other cells when they're expelled from the cell. Especially the chemotaxis, for example, is affected by glycogen before. It's a very strong chemoattractant, but also in the same cells, which leads to the cortical contraction, moving and bleeding. The pathway here is less defined, but there are some kinases that might be involved and been shown by people in the zebrafish and other settings. So we were looking at the possibility of this enzyme being involved in the pathway from the aquaporin down to the sensing of that mechanical stress and moving them macrophages from the meniscus area to the lining area. The first of all, what we did is looking whether CPLA2 is actually expressed and in what macrophages in the joint, and whether if it's expressed, it's in the perinuclear space where it acts as an activate when it's activated. And here it's in, you can see it in orange, together with a lamin, which would label the perinuclear space, in green. And if you look at the area around the meniscus, where most of our aquaporin positive macrophages are, you can see quite a good number of cells expressing CPLA2 or activated CPLA2. There are rare cells in the lining. If you start looking in this location in yellow, there's only maybe one here. And there are no cells in the sublining, which is less strained than the other areas. So we kind of think that also quantifying it, the CPLA2 is specifically favoring an expression, its activation is in the those aquaporin positive macrophages in the mechanostrained areas of the, say, meniscus here. So if we remove CPLA2, and here is a cross of CX3CR1, another macrophage driver with the FLOX CPLA2, here you can start seeing that actually they sort of resemble what's been happening with the aquaporin deficient mice, which means that the area that was covered by the aquaporin positive cells, that's nicely organized, becomes less organized, and there are more cells from the linings that you normally don't see in this area that appear in this area. So the kind of phenocopy is what we've been seeing in the other mouse model. And in fact, so this is a quantification of this phenomenon. And in fact, if you start looking in the more sort of zoomed out situation, where once again, the wild type CX371 control mice showing a beautiful, nicely organized lining here with the blue. That lining is somewhat destroyed in the mice that lack CPLA2 in the macrophages. So really phenocopying the what we see with aquaporin deficient mice. So the CPLA2 is enzyme that senses the calcium levels, and we see the differences in that when the enzyme is removed, but also we see that where the enzyme is located in, again, now it's a model system when the cells are taken out and they are wild type or knockout cells. So this is aquaporin knockout. The CPLA2 is coming like CPLA2 here in the wild type is coming in perinuclear space, but it's deficient in terms of its specific localization in the aquaporin knockout. We're trying to link the two together, and that is happening at the specifically at that constrained pressed 4 micrometer kind of chamber exposed cells. And similarly to what we see that with the migration that I showed you on the movie, when the cells kind of yellow and red cells moved around, similar with the CPLA2, we either the inhibitor of CPLA2 or knockout of CPLA2 results in the decreased migration in them. First of all, if cells are not pressed, and also if cells are lacking the CPLA2, kind of linking two together in one possible pathway. So just to summarize this part of the story, the migration of macrophages under confinement is dependent on CPLA2 activation downstream of aquaporin 1. Migration of macrophages under confinement requires aquaporin 1, and knockout of CPLA2 phenocopys knockout of aquaporin 1, and that leads to the this kind of pathways that we're suggesting, where the shell shape changes in response to the mechanical compression are important to sort of regulate the nuclear shape changing, whether it's swelling or stretching, activation of CPLA2, and the contractility of the actomycin that would be required for their migration out of the area and into that area where the lining macrophages are located. So with that, I'm going to put it all together into sort of summary of the presentation today, on both on the tissue level and the cellular level here.
Thank you, Irina. That was wonderful. Really appreciate you sharing these outstanding and unpublished results from your lab. Really exciting to go into this area that is quite complex, right? You want to conclude something more?
I just wanted to show I'm not sure which direction I'm going, the group.
Thank you, of course.
To thank, and especially the two main people in this project is Barbara Schoenfelder and Christina Deck, who was helping her.
That's wonderful. Yes, of course. Thank you. Thank you for everybody.
And our collaborators, obviously.
Doing this wonderful work, Irina. Really appreciate it, as always. An extraordinary tour de force and combination of genetics, outstanding microscopy, and mechanistic approaches to understand this complex biology, this specialized mechanosensing of these macrophages in very difficult area, right? The joints.
Yeah,
so
really very interesting.
Your efforts.
Every time it takes something takes us into completely new area of being very uncomfortable and trying to learn as much as we can very quickly, and that makes you tick. You know, that is when you become very excited.
And I guess this opens so many more questions, right? I imagine also in the context of knee replacements or as we age, I'm curious about the biology of these macrophages and the changes that may emerge.
Absolutely. But also, you know, one of the main, like, sort of general kind of conclusions out of it, we need to move, no matter what you have. You know, that's why they say, you know, whatever you have, the operation, surgery, anything to do with the joints, you have to move. You know, even if it's painful, you have to move. because then
Very,
it's
very impactful finding.
fast
Thank
to
you.
recover it, or like reconstitution of some of your cells, like we didn't know about for macrophages.
Thank you. So nice. It was a pleasure to learn from you. I would like to share in the last slide. Um,
Shall I stop
Now,
sharing?
let's see. Maybe I can do this. Let's see. You can see my other slide now, I think. So we can remind people that you can watch this Global Immunotalk from Irina that will be soon uploaded in our YouTube channel. You can also ask questions to Irina through our YouTube Global Immunotalk channel. Irina's talk concludes this first semester of 2025, but please stay tuned. We will soon post the upcoming schedule for the rest of the year. We are very grateful for our speakers, for our organizers, and for all of you joining us and continuing to learn together about the latest on immunology. So thank you so much. I will now stop sharing and we'll see you again after the break.